Hello, everyone, and welcome to a Facebook and Twitter live session with the very recently announced Nobel Laureate in Chemistry, Dr. Jennifer Doudna of the University of California, Berkeley, someone I have known for quite some time, and I am really thrilled to be able to do this Facebook Live, Twitter Live interview with her about the big, exciting story that the Nobel Committee decided to recognize about CRISPR-Cas and gene editing and all that. And Jennifer, thank you so much for being willing to spend 30 minutes with us in what must have been an absolute whirlwind since you learned of this particular award yesterday, very early morning. In fact, maybe I'll start there. I understand you actually didn't hear first from Stockholm. You heard from a reporter. How did that happen? And what were you doing at the time? Sleeping, I guess.
Yeah, crazy. Well, first of all, thank you so much, Francis, for inviting me here. It's a real pleasure. And you're right, it's a little bit of an embarrassing story, but I literally slept through a very important phone call and woke up when a reporter finally was able to rouse me. And I was just coming out of a deep sleep, and the reporter was asking, what do you think about the Nobel? And I said, I don't know anything about it. Who won it? I thought they were asking for comments on somebody else who won it. And she said, oh my gosh, you don't know. You won it.
I bet that reporter will not forget that moment either.
Probably not.
So ultimately, presumably, you did hear from somebody who was calling from Stockholm who kept trying, and they gave you the whole scoop?
As you can imagine, I immediately said, I need to get off this call and, talk to somebody official about this. And actually, my phone rang next with a call from Martin Yinek, who is the person who did the research in my lab at Berkeley, and he now runs a lab in Zurich. And so he called, and I knew it must be true at that point. And then I was finally able to connect with the folks in Sweden.
Oh my goodness. Well, some of us were watching live and wondering, is there an issue here? Is Jennifer off in the woods somewhere with no cell phone connection? But I'm glad that we got connected. So then it must have been like a pretty wild middle of the night there in California. And when did you get to talk to your co-laureate, Emmanuel Charpentier?
Well, we started texting. Crazily enough, we actually haven't connected by phone yet, but I'm sure her phone has been as crazy as mine. But we've been texting back and forth, and we're going to be talking later today.
Oh, that's nice. And presumably your email and your text inbox must be just overflowing with people sending words of congratulation.
It has. And, I have to say, that is so wonderful to hear from so many people. I actually heard last night from two girls who were in my fourth grade class in Hilo who still remembered me and who reached out from Hawaii. And I also, just before this meeting, this call started, I heard from my high school biology teacher, Marlene Hapai, who taught me biology, which was really nice.
Oh, that is really nice. Did she have something to do with your choosing this career pathway?
Well, she certainly was wonderful at communicating the joy of doing science. She had she was a very, very passionate, happy, active person who, wasn't telling us to read the textbook too much. She was actually into doing things in the classroom. So she really taught us kids that science is about discovery. It's about a process. It's about figuring things out. And I think, I really owe her that, thinking all the way back to, those high school days. I think I owe her that sort of passion for science that she communicated.
That is such a great message, Jennifer, because people who read about science or hear a lecture about science can get interested in it, but doing it, that's what really gets your excitement going. And it sounds like she did that for you. And lots of scientists have stories like that for sure. Well, Jennifer, say a little bit about this prize and what it's for, this gene editing CRISPR-Cas tool. Not everybody knows exactly what that is and why it's such a big deal. So say a bit more about what this is.
CRISPR-Cas it's a tool for genome editing. What does that mean? Well, it means changing DNA in a precise way in cells that can allow very tiny changes, including changing a single letter in the genetic code of a say, a human cell, all the way to cutting out genes or entire pathways and replacing them with some other kind of genetic information. And that's really what CRISPR enables. It's a tool for manipulating DNA that actually evolved a long time ago, probably long before humans came along, as a bacterial immune system. It's a way that bacteria fight viruses. And it's really through the process of understanding the mechanics of that and the biochemistry of that it was possible to harness it as a technology for something different, enabling genome editing.
So people may have trouble figuring out what the pathway is that leads to this kind of a development that wins a Nobel Prize. And it usually starts somewhere back in the basic science arena, maybe even in an area that seems a little obscure. What happened that brought CRISPR-Cas out of obscurity into the Nobel bright lights that we're talking about today?
Well, it is a great story, I think, Francis, because it really does highlight how science is actually done. And in the case of CRISPR, it's a field that 20 or 25 years ago didn't exist. Nobody had heard of CRISPR back then, and really started with a few bioinformatics labs that were noticing the presence of repetitive DNA sequences and the presence of little short pieces of viral DNA that were inserted between these DNA repeats. And those parts of bacterial genomes came to be called CRISPR. And in fact, it was my colleague, Jillian Banfield here at Berkeley, who brought these to my attention back around 2006, because her lab was doing a lot of the fundamental research on bacterial communities in natural settings, like in soil and water, that tend to have a lot of these CRISPR sequences. They probably have to fight a lot of viruses. And so from there, it was really, a handful of labs that had started to work on these in sort of the mid to late 2000s. And in fact, in those days, we had an annual CRISPR meeting at Berkeley. And when I was on the phone with Jill Banfield yesterday, we were remembering that back in those days, you could fit the entire CRISPR field in a small conference room at Berkeley, about 40 people. And that was it, and so from there, scientists started to understand a little bit more about how these pathways work. And they were very interesting. It was incredible that bacteria could literally learn about their infectious agents and store pieces of DNA from those viruses in the genome in kind of real time, and then use that information in the form of molecules of RNA, which is what I've always studied, to protect themselves from future infection. And so it was really that, basic understanding that allowed the system to ultimately to be harnessed as a technology.
So you could have, of course, worked on lots of other areas of research. You certainly knew a lot about RNA. CRISPR probably seemed like sort of a curiosity when it was appearing there, but somehow you decided you're going to put an awful lot of your scientific effort on that. Tell me why. What was it about this that you said, this is what I want to do?
Well, when I first started working on it, it was definitely, I thought of it as kind of just a fun little side project. And in fact, I felt a little guilty because, I had funding from the NIH and from Howard Hughes Medical Institute, and I thought, how can I really justify this crazy little project on bacterial immunity that might have zero to do with human health, but I just think it's interesting. But frankly, Francis,
we decided it's okay.
But frankly, the reason I think for me, I've always been fascinated by evolution and kind of questions of, where did life come from? And I really owe that to my graduate advisor, Jack Shostak, who was, thinking about that back when I was starting grad school in the 80s. And so in my own research, that's kind of always been an underlying theme. I think in the case of CRISPR, it seemed likely that, first of all, it was very surprising that that bacteria might have an adaptive immune system that would rely on RNA. And I thought that there might be a lot to learn about immunity in general, even in our own cells, even though it works differently. But if you could understand fundamentally how cells evolve immunity to pathogens, that this would be, just an interesting area of biology.
By the way, people who are listening on Facebook and Twitter, if you want to pose a question to Dr. Doudna, you know how to do it. Enter that into the system, and we will try to get to some of those here in a minute. I'm curious, Jennifer, though, just in the course of events here, so you're studying this as a really interesting system of bacteria having their own immunity, where they basically keep this little file cabinet to remind them what viruses they might have seen before and how to go after them. When did it occur to you that this might be more extensively applicable than that this same system might work in other cells, maybe even in human cells? Was that like a gradual realization, or was there a moment?
Well, I think it happened for a couple of reasons. One is that at the time that we were doing this research with Emmanuel's lab, there was a lot of research going on in many other labs, seemingly disconnected to what we were doing, that concerned either DNA repair in human or plant cells or animal cells, and using engineered proteins to trigger targeted genome engineering. So these engineered proteins could cleave DNA at precise places, and then DNA repair would take over and introduce a change to the DNA. And these were clearly powerful technologies, but they hadn't really taken off very widely, because they were, they required a fair amount of protein engineering and, just enough kind of, expertise of a special type that most labs couldn't adopt these very broadly. And so that was all going on, and I was aware of it because, papers were coming out in prominent journals, and I was, seeing these papers, and it was very interesting work, but didn't seemingly connect to what we were doing until we realized that the way the CRISPR-Cas9 protein works is that it takes a piece of RNA and uses that as the address label to cleave a matching sequence of DNA, and that it was trivial to change the address label by just changing the sequence of the RNA that this protein would bind to. And that was really fundamentally what we did with Emmanuel's team. And once we understood that, then we could make that connection and see how this could be a technology that could cut DNA and then trigger the kinds of repair that would induce genome editing.
So that was the big realization, that this goes even further than the bacterial you were studying, the system you were studying there. So at the moment that you were studying at that point, it looked as if this was primarily a means to cut DNA. A lot has happened since then in terms of how CRISPR can be used for other kinds of outcomes. Can you say a little bit about that? A lot of creativity seems to have emerged in terms of adapting this system.
Absolutely. It's been really exciting to see this work over the last 8 years, how the field has expanded in terms of even just the development of the technology. It all relies fundamentally on the mode of DNA binding that Cas9 or related proteins are using. It's this RNA-guided recognition, where RNA is able to invade the DNA double helix to form an RNA-DNA hybrid inside the CRISPR-Cas protein and allow the Cas protein to make a cut. And so, many clever scientists have recognized that you could use that fundamental mechanism and, decorate the CRISPR-Cas proteins with other functionalities that will allow them to do things like edit the DNA, meaning make a chemical change to a DNA base without actually having to cut it, make a double-stranded break, and go through the whole repair process. And there also are now ways to use CRISPR-Cas so that it doesn't cut DNA at all, but instead just triggers transcription, either to activate it or decrease it. So you can alter the output of the genome without actually altering the sequence of the genome. And these are the kinds of tools now that, scientists have at their fingertips. And I think we're going to continue to see expansion of that toolbox over time.
People often refer to this, looking for an analogy, as it's like your word processor program and you're looking at this enormous book of 23 chromosomes and 3 billion letters. And this is basically the function that says find, where you give it a sequence of letters of the nucleotides for RNA or DNA, and it goes and finds it. And then what you decide happens next is kind of how you engineered the CRISPR-Cas system. Is that a fair analogy?
I like that analogy. I think that's quite good. Yeah.
So in terms of applications, somebody's already asking, sending us a question. has CRISPR played any role in COVID-19, the thing that we're all surrounded by this year? Is there a connection there?
Yeah, great question. Well, it has, and I think it will in the near term for diagnostics, because and this is yet another sort of biochemical activity of a subset of CRISPR-Cas proteins that can be deployed for detection. And I love this application of CRISPR-Cas, because I think fundamentally it really goes back to the origin of the system. It really is about virus detection. And that's what these diagnostic strategies are doing, is taking advantage of that, that fundamental property, but rigging it so that the CRISPR-Cas protein, when it finds its target sequence, it triggers cutting of small reporter molecules that release a fluorophore. So you get a nice little visible signal when the CRISPR-Cas protein finds its target. So it's up, yep, COVID here, And so I think, there's all sorts of groups and companies and academic groups that are working on different ways of deploying that diagnostic, ranging from a laboratory test, kind of analogous to the PCR test that could run in very high throughput, but would require robots to do it, all the way to either a point of care test or even an at-home, home pregnancy stick type test that would use CRISPR-Cas. So I think, those are we're likely to see those over the coming months, and NIH has done a lot to, I think, really accelerate that line of research. And, and then I also think about it in terms of pandemic preparedness, having a diagnostic of that type, you could in principle program it to recognize any virus or any collection of viruses. So it gives us a tool that can be easily deployed in the future.
Yeah, I think those are really powerful. I'm glad we got to talk about them. People say with COVID-19 detecting the virus, well, all you need is RT-PCR. I'm going to geek out a little bit here for the people who are familiar with these technologies. The problem is, though, RT-PCR, you got to have a thermal cycler, and it's pretty hard to put that on a dipstick. Whereas what you're talking about, using CRISPR-Cas to basically find the sequence and then activate the cleavage of a fluorophore, you can do that at an isothermal temperature. You don't need a cycler, and that makes it a heck of a lot more amenable to all kinds of situations. So I think this is one of the more recently developed really cool applications that I wouldn't have thought of when CRISPR-Cas first came along. It just keeps expanding, doesn't it? Now, how about therapeutic applications, Jennifer, in terms of, is CRISPR-Cas going to lead to gene therapies that are going to save lives?
I think it will, with a little caveat that I think increasingly it's not about can this work, it's about how do we make sure it's affordable to people that need it and accessible. So I'll give you an example. So right now, and really this goes back several years, people appreciated very early on in the CRISPR field that you could use a technology like this to correct disease-causing mutations, especially for monogenic disorders like sickle cell disease, and lead to, essentially a cure at the source of the disease by correcting the underlying mutation. And so with sickle cell disease, we've already seen, incredible advances there. We've seen an announcement in the last year of a patient, Victoria Gray, who has been effectively cured of her sickle cell disease using CRISPR. And so that's, given the field just a tremendous boost in terms of thinking about the potential. And now what I think we need to do is really increasingly focus on developing the technology in ways that will make it accessible and affordable to those that need it, not just here in the US, but also globally. And so for sickle cell, right now, CRISPR has to be deployed by using a bone marrow transplant. So cells are taken from a patient and edited, and then replaced by bone marrow transplantation. Very expensive, obviously not an easy procedure, even though it's become more routine. And so we think about how to turn the technology into a much more easily delivered, easily deployed tool by developing ways to introduce the gene editing molecules directly into the cells that need it in the bone marrow without having to remove them. And that's one example of, I think, the kind of new kind of breakthroughs that are going to happen that will go hand in hand with CRISPR, but will really make this a standard of care for certain genetic diseases like sickle cell.
And maybe thousands of other genetic diseases where we know the precise DNA misspelling. And if we could just figure out how to program the editing system to go to the right cells and fix it, we might have a generalizable, scalable approach to all these conditions, which are very different in terms of their molecular biology, but they all have this commonality of a DNA misspelling. The challenge there, though, and I'd be curious to know what you think about this, maybe it's not so much the gene editor at this point, because that's gotten pretty powerful. It's the delivery system. How do you get it to go to the right cells really efficiently, make that correction in enough of them to have a benefit without having some sort of off-target problems? But gosh, shouldn't we be working on that? I guess we kind of are.
Big challenges, and yes, we should be working on that. I think a lot of people are, but right now it's not necessarily a coordinated effort. And as you know very well, Francis, there are some terrific delivery vehicles that include viruses, viral capsids that take advantage of their ability to hone in on particular cell types, but also really cool things like nanoparticles, even metal containing like gold nanoparticles that can allow encapsulation of the molecules that you're delivering, and various clever ways to try to label them for particular cell types. So there is innovation going on, but I think we need more.
Yeah, we have this somatic cell gene editing program in our common fund that's capturing some of that, but it could probably be expanded. This would be a great area for people who are thinking about science that they might want to work on. Oh boy, you could do a lot with this, but you could do a lot with everything we're talking about. And we'd be remiss, though, I think, Jennifer, if we didn't talk about the ethical issues that relate to gene editing. They've gotten a fair amount of attention, and properly so. Do you want to just say a word about that? And you've obviously written and thought about this and brought these issues to attention in various venues. What should we be concerned about?
CRISPR is, it's a powerful tool, as we've been discussing, and it was clear early on in the development of the technology that it worked quite broadly in many different cell types, including in embryos. Certainly animal embryos like mice could be engineered quite readily using CRISPR-Cas. And, there was no reason to think that it wouldn't also work in primate or even human embryos. And in fact, that's true. So that's been borne out by actual experimentation, both on non-viable and viable human embryos. And it's important to appreciate that type of that use of genome editing is, to me, quite distinct from what we're talking about when we talk about, correcting a disease like sickle cell disease, where we're making changes in a certain type of cell in a patient, but those changes don't become part of the germline, so they're not inheritable by future generations. Whereas changes in the germline, or in human embryos or sperm or eggs, these are changes that become part of the entire individual, every cell in that organism's body, and they can be passed on to the future generations. So, this is to me, quite a profound thing to think about when we think about editing the human germline and making changes that, if you took it far enough, would really change the course of human evolution using this tool. And so I've been really, for the last, at least 5 years, I guess, really actively working on this, thinking initially my thought process was quite, if I'm very honest with all of you, my thought process was, oh, I don't want to get involved in that. But very quickly, I realized that, as a scientist involved in the, very original work around this technology, that it was essential to be explaining it to people that are not scientists, not specialists, especially those in the government, in regulatory agencies, and to obviously lots of other people whose lives could, in principle, be affected by the technology. And then working to ensure that there was, at least among the international scientific community, some kind of common set of guidelines, or whatever you want to call them, criteria for proceeding for that type of, very special application of CRISPR. And honestly, I've been very pleased over the last 5 years. We've now had 2 international summits on this topic. We've had reports, including one just very recently issued from the national academies, that looked very deeply into this and made clear recommendations about how to ensure responsible use and importantly, transparency as scientists are working around the world. And so, you know, it doesn't guarantee, there's no guarantees, I guess, but I think this is a really good direction that we're in right now, where there is really a feeling among the international scientific community that we need to use appropriate restraint and stewardship of this powerful tool.
And you've been a real leader in getting that message across. And I think we all owe you a debt of thanks for bringing this to people's attention early on and getting those conversations started. And people interested should look at the report that just came out from the National Academy of Sciences and the Royal Society, because I think it is a pretty nuanced approach to this, scientifically sophisticated, and makes some very strong recommendations about the cautions that we need to pay attention to. Jennifer, I got to ask you also, this has been a remarkable week, and one of the most remarkable parts of it was yesterday, the Nobel Prize in Chemistry, a field traditionally dominated by men, was given to two women, you and Emmanuel. And many of us were particularly cheering for that feature. Well, how does that sort of set with you in terms of being a pioneer in your field in yet another way here in terms of this kind of recognition? And what would you say to women scientists on the basis of your experience of receiving this kind of recognition?
I guess I would say I'm proud of my gender.
You should be.
And may this be a you know, a sign of the times of change, that we will see many more such awards and recognition going to women where appropriate for their work. I think it's really, really important for women to, and girls especially, who are, you know, thinking about a career in one of the STEM fields, to feel like they are welcomed into the field and that they are their work will be appreciated. And you know, I think you know, there is a feeling among women, I think, in some cases, that there's a sense that no matter what a woman does, her work will not be recognized the way it would be if she were a man. And that is, you know, we clearly need to change that. It needs to be the case that we are all working together in a community of scientists, and we come from, you know, we are of different genders, cultural backgrounds, you know, personal circumstances, et cetera, et cetera. I have always viewed that as a strength in science. And I've seen, you know, I've now been running an academic research lab for 26 years. And I've seen over that period of time that the more diverse my group is, the more international, the more we welcome in people from all over, the better our science is, because people bring new ideas. It's the source of innovation and creativity. So that's one of the things that I treasure about doing science. And I just want to continue to see that broadening of the community of scientists, because I think it'll make science better. And it's, of course, the right thing to do.
It absolutely is. And you've smashed a few more holes in that glass ceiling, which has been there for too long. And really glad that this is a possible outcome among many others of Nobel Prizes this week. We only have about 30 seconds left, and I know you have a hard stop to do something else that's probably really important, and thank you for your time. Any last words of advice to young investigators or people thinking about becoming scientists as they contemplate the path that you've gone? What advice would you give, just as a bit of a final word?
My advice is go for it. Don't let anybody tell you you can't do it. You know, don't let naysayers dissuade you from what you are passionate about, because I think that is, you know, again, in my own personal experience and with people I know in science who I've either trained or worked with who are highly successful, it's because they trusted their inner voice and inner gut about science.
I love it. Well, Jennifer, you know, I've been fortunate to be a friend of yours for many years, and I'm so thrilled with this particular choice by the Nobel Committee. And thank you so much for spending 30 minutes with us, with lots of people listening. We'll tell you later how many there were, but there were a lot. And I'm sure they were benefited by hearing your words about how this all came to pass. Congratulations again, and enjoy all of this. It's going to be a wild few months, but just have a blast.
Thank you so much, Francis. It's been great. Thank you.
All right.